All right, folks, I think we're going to go ahead and start. So my name is Evelina Vasenko, and today I will be presenting a Release 48 webinar together with Omar Harb, who's located at the University of Pennsylvania, and Sim Rand from the University of Notre Dame. Today we will walk you through the various updates, new cool data sets and features in the current release, and also demo some of the searches associated with these data sets. Before we begin, I'll walk you through very quickly through the main landing page for the VEuPathDB site. And you should be able to see now the main landing page for the VEuPathDB database. So the database has several sites, and we will walk you through the searches using FungiDB, PlasmoDB, or VectorBase. But the neat thing about this that all databases essentially have the same structure, so that if you know how to use, for example, VEuPathDB, the same components and the searches will be available on other sites. So really quickly, the main landing page has a banner. Within the banner, you have a site search where you can search for a gene ID or a text term, and we will return essentially an itemized, detailed result of anything that has a mention of that term. You also have a series of menus at the top, for example, that allow you to navigate to your strategies, the specialized searches available here and also on the left. And these are different types of searches that you can initiate using the integrated data in VEuPathDB. And for example, if you're not sure under which category a specific search is located, you can always search within the filter to identify, for example, I would like to find a search that uses a list of Gs, a list of gene IDs. And so if I type ID, that's the search that comes up, so you can click on it to begin your search. And also you can access various tools such as Companion or Galaxy, access My Workspace to where you can analyze your data sets in Galaxy and bring them back to VEuPathDB sites for further analysis. Learn more about the type of data that we integrate and various data type summaries. Learn more about us. How to cite us, or for example, how to submit data. We also have a help section that allows you to access, for example, all learning materials that we provide, including information about current, upcoming, and past webinars. For example, if you're interested in learning a little bit more about what we have presented in the past, you can browse previous webinars and access recordings, as well as various support information. And if you have any questions, feel free to contact us by clicking on the Contact Us link. That will redirect you to a forum where you can specify or share the information with us. So coming back to the main landing page, I also want to point out that we have a section with banners, and some banners can be collapsed. For example, if you want to close, you can click on the X button just as I've shown you, but you can also bring this back by clicking on this reopen button at the top. This banner normally is something that we want to communicate with you, for example, in this case, register for the webinars. And finally, we also have a banner that tells you that the legacy sites can be accessed at the links, and at the links listed here. And also we provide a sort of an open question form for you to send us any feedback that you may have on the beta sites. And finally, just to wrap it up, I also wanted to point out that the specialized searches are available on the left, which is a section, as I have shown you, also accessible from the top menu. And within the center portion of the page, you can access various cards. They're designed to get you started very quickly with the searches and give you an introduction of how things work. You can also find more tutorials and exercises within the section at the bottom. And if you're interested in learning about news and various tweets that generated from our site, you can access the panel on the right, and this panel is collapsible. So once you have learned about various, for example, new data sets in fungi or GRDADB, you can collapse it further. Okay, so at this time, I will give the control of the screen to Omar, who will walk you through some of the recent updates that occurred in our site. And Omar, I will assign the screen share to you. Thank you.
Okay, great. Thank you, E, for the intro. I think everybody should be seeing my screen. We're looking at the VEuPathDB homepage. And as Gloria mentioned, we have a news and tweet section here on the right, which is normally collapsed. And so if you open this up, you can see all the news and tweets. This is if you're on the main VEuPathDB site. For each of our component sites, so if you are, for example, a PlasmoDB user, I'll take you to PlasmoDB, and on the PlasmoDB page, you will see the same structure, obviously, of the page. Everything is built on the same infrastructure, which is really nice for you. So you can, if you need to move from site to site, you can do that easily. And again, you'll see the news section, but now the news obviously is specific to PlasmoDB. So the first thing I'm going to demo to you is some of the new features that we added for this release. So probably over the next few months or next few releases, there'll be new features added regularly, and we will demo them as we give you these release webinars. So to demo this, I'm going to actually move to a site called PyroPlasmaDB. So that's one of our sites for Babesia and Theileria. Again, it's built on the same infrastructure. So all the features and tools that you are made available in one site will become available in other sites. And so here's a list of all the features. Specifically, first feature is that in the sequence section now, we have a way for you to easily copy a gene sequence from the gene page from the sequence section and to a clipboard. And I'll demo both of these features on PyroPlasmaDB. The second feature is on the gene pages, we also have genome browser views. And now we have the ability for you to easily scroll and zoom within the window without having to jump to JBrowse if all you want is to quickly peruse the region. And then two other features which I will demo. One is we now allow you an easy way to provide a screenshot with the contact us link, and and we made more prominent the buttons for downloading data and working with results, and I will demo this as well. This last one probably is not that relevant to you. You'll see errors every now and then. Those occur. Hopefully, most of them on a page reload, they will go away. But the error message that you're getting hopefully is a little bit more informative, and also it helps us capture any errors on the site so that we can fix them more easily. So for now, let me go ahead. I'm just going to find a gene page. So I'm just going to quick type the word kinase in the site search at the top here. And now when I run this, you will notice you'll get a bunch of results. Those are categorized here on the left. So you can, if you want, say I really only want to see genes. And so I can click on genes and that will show you only the genes results. I can also filter on these more closely and even filter based on organism. A question that has come up frequently is that, oh, you know, there's all this data and I need to go in and find the organism that I'm interested in. And it's really hard to sort of open up these submenus and then keep finding, you know, clicking to find the organism interested in. Well, the nice thing about this is that you can easily filter here. So I can, for example, start typing bovis, and now immediately I can filter down to Babesia bovis, apply my filter, and now my results are only results from genes because I filtered on genes for Babesia bovis. And so now I can easily click here and go to this gene page. So this is the gene page for adenosine kinase in Babesia bovis. And I can go down, and the first thing I'm going to show you is the sequence section. So if we go down to the sequence section, most of these sections are collapsed by default. This helps the gene page load much faster. And so again, if you're ever on a gene page and you're looking for specific data, it may be collapsed. So make sure you click on this little arrow here so you can see the underlying information. And you will notice now that there is a copy to clipboard button available with each of the sequence panels. And so I can easily copy, for example, the predicted RNA sequence if I want, or the predicted protein. If I click on this, it'll put it in my clipboard, and then I can go to whatever favorite text editor you have, and I can paste it. And so it gives you the sequence in FASTA format. And so that's a nice way if you need to take this to another resource so you can analyze it using other tools. This allows you to get the sequence very quickly in FASTA format. So the other feature I wanted to demo is the ability to scroll within the genome browser views that we have within a gene page. By default, the scrolling is disabled. The reason for that is that when we first installed this, we had it without scrolling was available. So as soon as you start scrolling down the page, this window, as soon as you move over it, it starts scrolling around, and so that was a bit annoying and confusing. But in this case, obviously we're looking at adenosine kinase, which is right here from Babesia bovis, and so this is the gene. You can see the direction of the gene. It has a little arrow on it. And also there's a color indication. So blue genes are going from left to right, red genes are going from right to left. So you have two indications, the color and the arrow. And now let's say you decide, well, I really would like to see what's upstream of this area here. And so without having to go to the genome browser, I can click on this button here to enable scrolling and zooming. And now I can drag it and it'll start loading, and I can view other regions on this gene. I can also zoom by double clicking. And so if we go back to our adenosine kinase, for example, or it doesn't matter, any gene basically, I can double click. And when sorry about the background noise, when I double click in the area here, it'll zoom in. And so I can keep double clicking as much as I want basically and view the gene closer. And if I want to zoom back out, I can click on the shift button at the same time and then double click on the screen. And so now what happens is I can zoom out. Okay. All right, so the next feature I will demo is if you run a search. So let me go back to my my searches. So a really nice thing, you could, if you want, you can click on the back arrow a few times until you get to the search of interest or to your previous search. Or if you notice here in the search box, there is a little arrow which allows you to go back to your previously run text search. And so this is a nice, really nice shortcut. So I'm going to just click on this arrow. So this takes me back to my search for kinase. And remember, I had filtered based on genes and also based on Babesia bovis. And so I'm back to my same search with the same filters. I can always clear my filters if I want. Up here, I can clear all of them. But in my case, I'm interested in let's say I want to download or work with these results. So I have 287 genes that came back from my search. So I'm going to click on this export as a search strategy. And you notice here you can download or mine your results further. So I'm going to click on export. And oops, let's see. All right, so this will export it to a search strategy, which will appear. Let me try this again. There we go. All right, so there are 287 genes. And now the new thing that we added in this release is these buttons, instead of being, there used to be small links right here. So now they're more prominent buttons. I can click on download. And while I'm looking at this, I'm going to show you a nice feature in the download section where, for example, I'm looking at 287 genes, right? And let's say I only want to download the upstream sequence genomic sequence of the genes because I want to look for promoter elements, for example. So I can click on FASTA, which is the sequence format, and now I can manipulate this and say I want to start at the ATG, for example, and I want to be upstream of the ATG, so we're going to make this minus. And there's a lot of information on the bottom here that helps you with this, so obviously don't worry about remembering this by heart, but I can say 300 nucleotides, and I'm going to stop also at the start codon, so I just want the upstream 300 nucleotides, and then I can click on get sequence, and this will provide me the upstream sequence from the 287 genes in one shot. So that's a very nice nice feature. Okay, so let's go back to our strategies. Okay, and then you can add items to your basket as well in using this button. And you can add additional columns. And so if you weren't aware of this, I can click on add columns, and then any data that's available in the resource, you can just open them up and let's say I want interpro IDs, if there are any. So obviously will update this column, a new column will apply, and in this case, these do not have any interpro domain, so it just says NA, but if there were any, you would see numbers here showing up. Okay, and the last feature I wanted to tell you about is the ability to quickly add a screenshot to the Contact Us link. So the Contact Us link is very important. We get many emails a week on a weekly basis from our users, asking us questions, asking for help, providing us feedback on the site, letting us know about data sets that they think we should be loading in the resource, and also reporting bugs or telling us about things they don't like, which is quite useful for us and helps us improve the sites. So if you click on Contact Us, this will open up the Contact Us form, and you will notice here now you can add a screenshot. And so I'm just going to quickly take a screenshot of this window here. So that's in my clipboard. So everybody, obviously your computer and the system you're using has different ways of capturing screenshots. So then I can click on add screenshot, and now I'm going to just paste it into my window and click OK. And so now I can provide information and say this page has a typo, for example, right? And so you can send it to us and you have a screenshot, and so that helps us then improve and fix things if you notice any issues with the page. Okay, so I think the next thing, so these were all the new features that we've loaded, and again, because our sites are built all on the same infrastructure, all of these features are available on all of our sites. Are there, let me see if there are any questions so far. Don't see any. Okay, so the next thing I'm going to take you is to start talking about some of the new data, and we're going to go back and forth between everybody on the call. So between myself, Sam Rund, and Eve, we will show you some of the new data sets. And so the first website we're going to be talking about is VectorBase. And so I'm going to take you to VectorBase. I was playing around with VectorBase earlier, so let's go back to the homepage. And again, if you go to the news section, you can go to the release, and then you can see here a listing of all the data sets that were loaded. In particular, what I'm going to talk about is the omics data sets, and then Sam will take you through some of the population biology data, which you see that there are many studies that were loaded in our MapView feature or tool. So we've added 15 additional microarray data sets. So remember from the legacy vector-based site, there are many data sets that we're porting over, and some of these are still being ported over, and I think these are all of them. And so we've added an additional 15 microarray data sets and also many VCF files, which include the genetic variation data from various studies as Genome Browser tracks. So you may ask, well, you know, how do I get to these data sets? So one way is through the news, and I can go to the actual search. So if I click on this to identify genes based on microarray, this takes me to the microarray search page, and this will list all microarray data sets available in VectorBase. And you will notice all the new ones have a new button, and this stays up for one release cycle. And so you can quickly see, oh, these are all the new data sets that are available. Another way is going to the data section up here in the upper menu. You'll see that there's a data section, and this shows you data sets in VectorBase. So I'm going to click on this, and this will list all available data sets. So there are over 500 or there are 513 to be precise data sets that have data set records in the database. Of course, this is all data sets, but you're only interested in the latest release. And you can look here at the top and you'll see this is release 48. And so I can type 48. All right, and then this immediately will filter the results. So now I'm looking everything, and you can confirm this by the column here, looking at release. And you'll see here, these are all release 48. So that's the latest release. I can continue adding, and I can start typing microarray, for example, and you'll see here that it starts filtering. And then I can keep doing this. You'll notice that the category here, they're DNA microarrays. This is the one that I was talking about. So I can just go ahead and type DNA as well. And now I've filtered, and it shows me all data sets that have the word microarray. I'm doing more of a general filtering here. I could click on this arrow and specifically say I only want to filter on the release number and the category, so you'll get a bit more refined results. So it really depends on what you're trying to find, but this is a quick way, an easy way for you to find what are the most recent data sets in a particular release. Each data set has its own data set page, and so this table shows you everything, but you can click on any of these, and when you click on that, you can go down and you can see more information, including a link to the publication if available. We do have data sets that are unpublished, and so they won't have publication, including a contact person, information about the study, and an example graph of what the data looks like, and links to the searches. Okay, so I'm going to take you and run you through an example or one of these searches. So I'm just going to go to searches, select genes. I'm interested in genes based on transcriptomics and microarray evidence. So that's going to take me back to the same page we looked at earlier. So these are all the data sets with that are listed as new. And let's go ahead and just take a look at these and find something that looks potentially interesting to search on. So there are a bunch of insecticide ones, and it really doesn't matter which one we do. Let's take this, maybe this life cycle one. And I'm going to select a fold change search. Okay, you'll notice here there are different types of searches available for the data. It really depends on the type of data and what kind of searches we can activate. And so for this particular data set, we have a fold change search and a percentile search. So both of these could be potentially useful. And so in this case, let me go ahead and select, I'm going to compare the midgut to the head. And I'm asking for any gene that is up or down regulated, so differentially regulated by twofold. This gives you an example gene that should be returned. So basically, we're saying the twofold will be in between these two samples that I selected. And then you click on Get Answer. And so this will return results. Hopefully, there are some. There are. So there are 145 genes that are differentially regulated between the midgut and the head samples that were analyzed in this particular experiment. And then you can go ahead and take a look at them down here and analyze them further. You can add more steps. So the beauty of the strategy system is that you're basically doing an experiment. So I can add another step here, and I can say, well, out of these 145, do any of them have predicted secretory signal peptides, for example? And so I can click on the Add Step button. It tells me I want to combine these with other gene results. I'm using an intersect operation because I'm trying to find things that are present in my 145 and also have a signal peptide. And now I have to choose the signal peptide search from this list of queries. Again, you can try and find where the query is, and hopefully the categories make somewhat sense, but it's often much easier to just simply start typing signal peptide, and it'll filter your searches. Now I can select the signal peptide search. I'm just going to select all of these and run my search. And so now this is going to run a search in the background. It's looking for all genes that have predicted secretory signal peptide. There were 78,000 in VectorBase, as expected. 26 of them are also found as differentially regulated in this experiment, and then you can take a look at them further and analyze them further down here. So this is as far as the microarray data goes. I'm going to go back to my dataset section and datasets in VectorBase. And now I'm going to look for a different type of dataset. The other dataset that I wanted to describe is the VCF files, and just show you where they are and where you can look at the genetic variation. So I'm going to go ahead and select again. I'm looking for 48, release 48, or you don't have to limit by release. And immediately you'll notice there's a category here, genetic variation. So this is the one that I'm actually interested in. But you can start typing variation, for example, and now you'll notice that all of them are filtered. Then you can obviously make it more specific to really fine tune what you want. And now I can click on any of these studies, and you'll notice again, each data set has its own description, and you can get more information about it. But this data is loaded as genome browser tracks, and so I can go to my tools section, and let's say I want to specifically look at this data set. I can go to tools and go to the genome browser. Oh, sorry about that. Get back here, genome browser. And now when I go to the genome browser, it's going to load the default organism, and give it a second to load. While this is loading, see if there are any questions that we need to answer right now. I don't think so, but you'll let me know. So I'm looking, in this case, I'm looking at, this is Aedes aegypti, I guess, in this case. And I can click on any gene, obviously, and get more information. And so let's stick with Aedes aegypti, just for the sake of time. I can click on select tracks, and I'm interested in genetic variation tracks. So right here, there are nice categories that are, there's a huge amount of tracks, right? So you can't expect anybody to know exactly where to go, but you can easily click on this genetic variation option here, category, and now all the genetic variation tracks have been filtered. And now I can select them all or just select a couple of them. Let's just select 3 of them. As you're selecting them, they start loading in the background. And now I can collapse this, and you'll see here now the data from the VCF files from these studies have been loaded. And now I can scroll down and look at them further, or move around and analyze them. Each of these SNPs or variant has more information, so I can click on it, and this gives me all the information that's coming out of the SNP analysis that was available to us to load for the release. Again, you can zoom in out. Yes?
There is a question about RNA-seq data. Someone is interested in looking at the coverage. For example, could you zoom in to reads to where you can potentially look for SNPs using RNA-seq data, not the SNP data that you have just shown?
Right. So for SNPs, for RNA-seq data, we don't load the BAM files, which include the actual read level data, because they're huge amounts of data sets to store. So we load them as big files, which unfortunately that loses the actual read level data. And so we can try and take a look at that here. So let me clear the filters, and let me pick, in this case, we're looking at RNA-seq, so you can filter by the category RNA-seq. And you'll notice here there are many different types of RNA sequence tracks. So once you find the one that you're interested in, either you can filter or you can find it, but you'll see here there's a coverage track. So if I click on this will load a track for the coverage. I can go ahead and close the SNP tracks, which I'm not interested in right now. And then from this particular experiment, I can start seeing what the coverage is, but unfortunately you can't zoom in to see the tracks themselves from RNA sequence data. We do have in our workspace, we have a way for you to analyze your own data using Galaxy. And so if it's something you really want to do, you can do this in Galaxy. We have predefined workflows for you, so you can bring the FASTQ files from the sequence read archive. We already have preloaded all the reference genomes, or all the genomes in VectorBase are preloaded. Actually, all the genomes in all our databases are preloaded, so you can select Aedes aegypti, and you can realign the sequence. You don't even have to go through the whole analysis. You can just do an alignment using, whatever alignment program you want to use, and then you can get the reads. And in fact, you might be able to use other analysis tools which will help you define the SNPs instead of you having to go in and do it on a case-by-case basis, looking at the reads themselves. So my recommendation would be for you to, if you are interested in really trying to extract SNPs from RNA sequence data, is to go through the trouble of reanalyzing the data, because there will be tools that allow you then, based on the alignment, to determine SNPs and define some of the robustness of the SNPs. Because if you have one read that has a nucleotide change, how much are you going to trust that, right? So you want to be able to do something a bit more statistically robust. And having to do this, you can imagine a gene could have 1,000 reads or more aligned to it. And so you're going to have to try and look and count the reads. And so that becomes, I think, very laborious. And so my recommendation is for you to reanalyze and then use the appropriate tool for determining SNPs based on the aligned RNA sequence data. So hopefully that's helpful. Okay, so there's another question. Seems that GRDB has less information. Is this because interaction do not? Yeah. So the less information, I guess by less information you mean potentially less data, and that really depends on what's available to us. And there are a number of data sets that are still remaining to be loaded. I know there are a number for Giardia, a number of new genome sequenced or resequenced, and so definitely those will be coming. We do rely on the community to let us know. So I think this was from Maria Tools. Welcome to the webinar. So please definitely send us a contact us link from any of our sites. And if you notice that there is clearly this data set from Giardia that has not been loaded, that should be loaded, let us know, because the assumption is that we know about all data sets, but that's not true. And so you telling us about a data set is quite informative and will help us make sure that we have it on our tracking list and are making it available and for a future release. Okay, so I think in the interest of time, we should switch over to Sam, who's going to let us know about, I believe next up is Sam, right? To let us know about the population data loaded in MapView for this release of VectorBase. And so Sam, I'm going to make you the presenter.
Thanks, Omar. Yes, good morning, everyone. I'm sorry, I've got I've had everything all set up in the wrong screen. Yes. So I'll be showing you today MapView, which was previously known as PopBioMap in VectorBase. And this is presently only available in VectorBase, but we are working to bring it to our other sites. And as you can see and guess from this map, all the data is geospatially and temporally described. So for this release, a very big release, we actually have released 45 new MapView projects, which you can see here in our news release. And for one kind of our data, our abundance records, we actually added 219,000 new abundance records. And it's also our largest increase in pathogen status assays, which we doubled our records to over 174,000. And as you can see from this list, some of these are from abatement districts like regular mosquito surveillance in Manatee County. And for many of these, they've deposited data in years past, and now they've deposited more, often perhaps the 2019 data, or sometimes they went back further and the depositor saw the data that they deposited for the first time, and so they went ahead and sent 10 years of historical data. One that I want to highlight of particular interest is data from the Ugandan isomer site. And as you can see from our view in this project, both pathogen status data was provided and curated, as well as abundance data. And so first I will switch using this view switching screen to see our abundance data. And when I do that, I can click on a data point. We can see a graph of the data over time. So you can see the abundance of Anopheles mosquitoes over time. And you can change the resolution of that data to see at a finer resolution. You can use the sliders to zoom into a particular time of year, or even make it bigger so you can get a better idea of what's going on. Next, I'll use the view switching screen to switch menu to switch to pathogen view. And so for this data set, we also have the testing for malaria from the collected mosquitoes. And as you can see, the number of malaria detection assays that were performed, so it's probably a dissection, can be seen using the bar charts, using the scale on the left, and the number of positive Plasmodium falciparum infections is depicted using the line chart with the axis on the right. And I can actually click here on list details, and I can actually see exactly how we have all the metadata. Oh, actually, for this data, we didn't record exactly how the pathogen status was determined, but I'm assuming it was dissection. And I would like to mention that we've added 6 new Anopheles gambiae complex abundance projects for the African continent with this release, and we are looking to add more. So if you have any seasonal abundance profiling data, especially if it's been resolved to the subspecies level or the cryptic species level, please do get in contact with us. I'll next highlight some data from a U.S. mosquito abatement district. This is Ada County. Right now we don't see any data on the screen because we have these filters here. And I know that Ada County does not use New Jersey light traps. So if I remove that filter, I will suddenly see all of their trapping locations with data. And I can click on a location, adjust temporal resolution, and see that plot of the data. And as I said, we have over a decade's worth of species resolved trap counts. Now I can also, just as I removed a filter, I can drill down and add filters to make it more restrictive. So let's say I just want to see Aedes species data. And I can get that. And I can also use these faceting on menu here on the right. And I can, for example, restrict to, let's say, I just want to see what was caught in one particular kind of trap. So I can click there, and that adds the EVS trap catch to the filter bar. And note, as you click on data points, you can continue to zoom in, and some of these data points will become more data points as we zoom in. So we really do try to have trap level data. So if you notice when I double clicked, that one point actually represented 6 different traps when I zoomed in. Here is a new Aedes aegypti insecticide resistance phenotype assay in Jamaica that was submitted. And notice in this view, when we click on a data point, we get different kinds of graphs. So instead of showing us obviously abundance over time, this is showing us the data on the insecticide resistance phenotype assay. And as you can see, we have all the metadata. So we can see that, for example, this data point was collected by exposing mosquitoes for 24 hours to 0.016 ppm of methaprin using a WHO larvicide assay, and 53% of the mosquitoes die. And also in this insecticide resistance phenotype view, you can see that the colors of the dots represent the degree of resistance. And as I mentioned in the other view, you can see all of the metadata that we have. You can, for any view, download all the raw data. So these are the fields, for example, in this view that you would be the columns in the CSV download. And I really like this feature. You can click on this share link button, and then that will copy a link to your clipboard, and then if you send that link to a colleague, for example, and they click on it, they will see what you see here on the screen now. So it's a very good way to work with collaborators. And finally, we'll switch to, I'm not going to talk about blood meal status. There's no new data sets this release, but I will show genotype, which presently is just insecticide resistance genotypes, but as we expand across our sites, we'll be adding other kinds of genotype locations. And you don't see anything right now because we still have that filter for a project that doesn't have any genotype data. So I'll remove that filter, and you'll see data that we have from all over the world. And I'm just going to quickly click on this. I'm going to go to a locus, I'm going to click on our knockdown resistance locus. Is one of the most commonly surveilled locus. I want to just look at species, so we'll just do A's aegypti, and by faceting by allele, I can click on an allele, and here the graph that we have will show you the breakdown between the number of organisms that were assayed that had the wild type versus the resistant mutation. So that's all that I want to show you today. Please do stay tuned. We're working on a pretty significant revamp of the map right now that will allow us to roll it out to other parts of the site. But if you have any data sets, not even just in vectors, but in the different component sites that you think would look particularly well visualized in this mapping geospatial temporal interface, please do reach out to us and let us know. Thank you.
Great, thanks, Sam. So I'll go ahead and take the presentation over and quickly highlight a couple of additional data sets that we've loaded in TriTripDB and PlasmoDB. And so I'll try and be quick so we can get to some updates from FungiDB. So again, I'm looking at TriTripDB. I have the news section open up. Let's go ahead and take a look at this. And you'll see here that we've loaded this ChIP-seq dataset from 4 histone variants. And this release, we mapped it to the new T. brucei lister 427 2018 assembly from Nikolai Siegel's group. And you can get information. I can go to this page and I can look at it. And so I'll show you this in a second. But before I move away from the news section, you will notice that we have information about updated annotation. And so we have, on a regular basis, we update the annotation of our genomes. And as some of you know already, we are working on implementing Apollo for, which is a way for you to update annotation as well. And that's something we can then do on a more regular basis, update user-provided annotation. And so that will be coming available very soon for all of our sites. It's already available for many of the vector genomes. And if you need to get more information about, well, what was updated, you can see that there are updates here, but you can always click on the summary here, and this takes you to a spreadsheet, which gives you all the information that you want to get about the genes that were updated. And this is true for both TriTripDB and PlasmoDB for this release. You will see a same section in each of the news sections, which give you information about the updates. And of course, these updates are made available on the gene pages as well. So for this particular data set, this is just a ChIP data set. There's an echo. I think, Sam, maybe you should mute yourself. Or somebody, sorry about that. Okay, so I'm in the data set for this histone ChIP-seq section. Information about it, the publications as usual, description of the data set. And you'll see here at the bottom here, there's a link to the genome browser. So instead of trying to go and find it, you can click here. This will load the genome browser in TriTripDB, and it will load the track for you for this particular data set. And so as this is going on, you'll see here, I've collapsed my genes. So this is a nice feature in the genome browser. You can click on this on any track, and you can select display mode, and you can do either collapsed or normal. If I do normal, you'll see that the genes will pop up like this. The nice thing about the collapsed view is that in the case of TRIPS, you can easily see that the strand switch regions, right? And you can see that this is a density plot of all the ChIP-seq tracks, and where it's darker, it means there was more alignment of reads. And so that's nice. You can see that these reads are aligning to the strand switch region. And if you mouse over the track here, you will see which modified histone was being recognized. And so you can see that H4 is, for example, showing up there. And you can move around, obviously, and you can move between chromosomes if you want us to look at, let's say, chromosome 2. And so you'll get a same view here, and you will see the density plot for this. So quickly switching to PlasmoDB. So again, same structure. So I can go to the news. You will see information about the recent updates to annotation, and you can explore these further. The new data set we loaded is a proteomics data set for amerosome data, and this is from well, publication is right here, and you can see it's from it's a recent publication. But the bottom line is that we allow you to search this data or view it in different ways. So we align the peptides, and you can look at them in the genome browser. But importantly, you can search them, right? And so down here you have searches available for this data set. So I can click on identify genes based on proteomics data. You can access the search from anywhere we have searches. So you can go under genes, tools, proteomics data, and then mass spec, and you will get to the same page right here. So different ways of accessing this. This particular data set was in Plasmodium berghei, so I can expand berghei, and I can select this one from Shears et al. This is from Fortini Sinus's group. And so now it's the merosome data set. I can play around with the number of peptides that I'm interested in. So I can say I want genes that only had 10 peptides, for example. So I can modify this to 10 and click on get answer. And so now this will return all genes that matched the criteria from my search. So there were 703 genes that met this criteria. And here are the peptides that mapped, and additional columns about this product description. As I mentioned before, you can add more columns if you want, or remove columns. And so that's really highly useful. Okay, so Eve, I'm going to stop here to give you some time to talk about, I know there's a lot of data for FungiDB, and so it's time to switch over, and I will monitor the questions and answer them as they come through. Thanks.
All right, sounds good, Amar. Thanks very much. All right, I am on FungiDB news release page, and as you notice, we have quite a few data sets in this release, including RNA-seq, ChIP-seq, SNP analysis data, and also updated functional annotation. Just like Amar showed you before, you can always access what type of functional annotation has gone into this release by clicking on the link, and we actually provide a sort of more detailed view as to what has been updated, for example, products, gene names, Go terms, PubMed IDs, etc. So today I will walk you through a few data sets that we have integrated for Aspergillus fumigatus, which is a pathogen that causes aspergillosis. And in particular, I will start with the search of the RNA-seq data for Aspergillus fumigatus that came from a study published this year. So I'll click on the dataset name to be redirected to the dataset record page. And as you have seen before, this is a dataset record page where you can access information about the dataset, sort of a brief summary about the samples, and also deploy particular searches. For example, I'll click on the differential expression, and that's the type of the analysis that is run on the datasets with replicates using DSeq2 pipeline, and I'll choose to analyze and compare a mutant that was grown in no drug conditions and mutants that have been grown with itraconazole. So the significance of these mutants is that a recent study investigated essentially transcriptomic changes that occur in fungal pathogens in response to treatment with azole drugs. And they have particularly looked at the negative cofactor 2 complex, NCTA and B, the specific factors important in the response to azole treatments. And so what we will be looking at is essentially comparing the upregulation in gene expression in the mutants when treated with itraconazole. I'll choose upregulated, and we will leave the full difference at 2, and I'll click on get answer. And so once the search returned the results, you will notice that the screen is being refreshed, and I'll just move this up to have it on my screen. Here we have about 692 genes, 90 genes that fit the criteria. And so from this point, you can either look at individual gene record pages, also look at the fold change and associated statistics for these data sets, view it in JBrowse, such as coverage plots, etc., where you can expand the strategy and create a sort of an in silico experiment. What I would like to do, I would like to I would like to add a step and bring in another data set that we have integrated, and that is specifically SNP calls on the whole genome sequencing data of isogenic strains from one patient. And what I would like to do is to take a look and identify whether there are any non-synonymous mutations that occur within the genes that we have identified here. So let's go ahead and click on Add Step. Notice we have several menus available, so I'll be looking under sort of not genetic variation, but SNP data for SNP characteristics. And by clicking on this link, I will navigate to Aspergillus fumigatus data, and the data are aligned to AF293 reference strain. And from the menu available on the bottom, I'll choose the specific data set that I'm interested in. So in this release, we integrated SNPs from 13 isolates that were identified and recovered from the patient. And I will look for those SNPs that are non-synonymous to return at least one or more, and leave the rest of the criteria at default for now. And so as the screen refreshes, we will see that we have now 3 genes that potentially may have some interesting SNPs. And if you scroll down, you will have essentially a list of genes with the non-synonymous SNPs. And also, if you want, you can activate the expression data for these genes as well. So what I can do, I can also cross-reference this data with a set of Aspergillus fumigatus knockout collection. And just really briefly, the study that we are looking at is published in this paper, and the study also produced a gene-wide knockout collection for a selected group of genes, such as transcription factors, and we have integrated this data in FUNGDB as well. So the CoFun collection has a phenotypic data, such as images recorded at various temperatures at different times, that you can also view in FUNGDB, on FUNGDB gene record pages. So I will show you a quick step how you can access this data. I have identified, essentially, I have a list of genes that we know are in CoFun collection, and what I will do, I'll navigate back to my strategy and say, well, you know what, how many of the genes that we have actually identified being differentially regulated when fungal organism is exposed to itraconazole treatment, how many of these genes actually present in the collection? So what I will do, I'll add a step to intersect with my gene list. So I'll click on the insert step before, and since I have a list of genes, I will search for an ID to quickly identify gene ID search. And in this box, I'll essentially paste the list that I have and click Run Step. And let's just finish the strategy. Like it will finish just in a few seconds. Here we are. I'll go ahead and delete this step for now. And so the observations from our analysis is that out of 690 genes differentially regulated with the treatment using itraconazole, 15 of those genes are actually in the CoFun knockout collection. And notice that I have mentioned before that we have run this analysis using a RNA-seq data set that was aligned to the reference strain AF293. However, there's also another strain in Aspergillus fumigatus, 1163, that is also used in lab conditions. And there's a quick way when you can transform your observations into another strain. And you can do this by clicking on adding a step and transforming your data into a 1163 strain. So I'll just search for fumigatus. Here we are. And so here, what I'm doing, I'm essentially harnessing the data that I have identified in the reference strain AF93, and I'm transforming my data into another strain. And that potentially is quite an important step, because we know that you may have a different number of genes, as we see in this result, matching to your search. That means that for one gene in AF293, there could be 1, 2, or 3 or more orthologs matching in the 1163 strain. And so to examine this data further and visualize the phenotypic data, you can click on individual gene ID links, and I have already pre-opened them before. And once you're on the gene record page, you can click on the phenotype section to be redirected to the section that presents the data for the co-collection. And by expanding the link, you'll bring up essentially an image that was taken at 72 hours when strains were grown at 37 degrees. And this, I think, I have shown everything that I wanted to show in this demo. And as always, if you have any questions or if you would like to suggest new data sets, feel free to contact us at the Contact Us link. Amar, are there any questions?
I don't see any questions. Maybe while we're waiting, if anybody wants to ask questions, just to point out again that we covered many different types of data and different ways of searching them. But for every one of these, we've actually already run a webinar over the last few months. We've run over 20 webinars so far. And if you go, maybe Eve, you can show this, if you go to the help section and go to learn how to use VEuPathDB, you can access this from any of our sites. You can go to the webinar tab, and this will list all the previous and upcoming webinars. So you'll see that the upcoming ones, we still have to, there are a number that we're still scheduling. So those will come back to this page. This will be updated soon. But you will notice as Eve scrolls down, you will see that there are a number of webinars that are really specific. So if you were confused about the transcriptomic searches, then you can go and do that. If you want to learn more about how to run a Galaxy workflow, you can go to the Galaxy one. If you're interested in proteomics data in VEuPathDB, you can go to the proteomics one. And those are all recorded and on YouTube. And of course, if still there's something confusing to you, definitely drop us an email regardless, and let us know what you think. So that will be incredibly useful. I want to highlight next week's webinar. If you're interested, we are having a webinar hosted by a sister resource called MicrobiomeDB. A number of people have been asking about, well, do we have resources for microbiome data? And so we don't necessarily have them within the NIAID funded resources, but we do have a sister database called MicrobiomeDB, which is funded by the Gates Foundation, and that integrates microbiome data from all sorts of conditions, not only from parasite infected individuals, and so or even animals or insects. So that would be a pretty cool webinar to attend and learn more about how you can take advantage of microbiome data available, and also some analysis tools that are available for you to analyze microbiome data. So I'd recommend that you register for that webinar if this is something you're interested in.
Right.
There are no additional questions.
Like,
yeah.
Oh, sorry. Yes. Yeah, I was just saying that it seems like that there's no questions. So I think we will end here. And thank you very much for everyone who have joined today.
Thank you. Bye-bye.
Bye.